This is the light microscopy research group session. So if you're in the wrong room, just stay because we're a bunch of cool people. We do really cool stuff. So this is going to be a little bit different. We're going to have a lightning talk first on EM, and then we're going to go into basically the paper we're currently writing, which is the research project that we're trying to finish up the writing for. And then if we have time, we're going to talk about the project that we're moving towards, which is AI. So if that sounds reasonable to everyone, without any further ado, Murray, take it over. And this is his first ABRF, so please be kind, no rolling, no throwing objects. We like new members.
Okay, everyone can see my slides? Yes, fantastic. Yeah, thank you so much to Richard and the organizers for this chance to do the lightning talk. As you said, this is my first ABRF meeting. Very much enjoying it so far. I hope this continues through the rest of the conference. So I'm going to talk a little bit about some work that we did over the last couple of years, which brought together a bunch of techniques that my core offers as like histology services, light microscopy, and atomic force microscopy, to look at a problem associated with pancreatic cancer. So I'm the director of the Molecular Cytology Core at Memorial Sloan Kettering. We offer a wide range of services. We offer histology support from embedding, sectioning, through to IF and IHC and FISH. We run the imaging facility for basic research, so confocal microscopy, other forms of light microscopy. And we also have a suite of 2 atomic force microscopes, which we use for mechanical characterization of cells and tissues, as well as single molecule imaging of proteins, DNAs, other molecules. And the wonderful thing about the AFM is that it brings together several of the techniques that we offer into one really neat package. And this slide shows a couple of examples from the last few years. The left-hand side shows some of the AFM measurements on a tissue section that you can see stained with Masson trichrome stain. And you can see the staining differences with a greater or lesser degree of collagen, which is this darker stain, as associated with a greater or lesser degree of changes in the Young's modulus, which is kind of analogous to stiffness. And then on the right, that was done with adjacent sections, but we can also do this with endogenous fluorescence as well, which is what you see on the right. This was work done in collaboration with the Elaine Fuchs lab at Rockefeller University. And so here you have a dermis sample where you can see very distinct stiffness differences that are associated with different areas of the derma and the surrounding stroma. And you can see this kind of line of green integrin alpha 6 in the image in the kind of top right of that figure. And then that's associated with stiffness differences in the bottom right. But the project, which I'm going to talk about in a tiny bit more detail, is something that we did in collaboration with Sylvie Debord and Richard Wong's lab at MSK. And they work on head and neck cancer and also pancreatic cancer. This is a very invasive cancer. It's it travels very quickly through the body, metastasizes really easily. And so they created a model using Matrigel and these microchannels, and you can see the design in the top left, to look at the interaction between Schwann cells, which are associated with nerve tissue in the peripheral nervous system, and pancreatic cancer cells. And it was known before that there was some kind of association that PDAC, these pancreatic cancer cell lines, preferentially moved along nervous tissue to invade new tissues. And so they created this model where you could view this happening as they travel through Matrigel together. And you can see that the Schwann cells wrap around the cancer cells, the green Schwann cells, and the purple cancer cell lines. And they help push each other along, and you can actually measure these kind of longitudinal waves. And it's dependent on reprogramming of the Schwann cells in a CJUN, which is an oncogene-dependent manner. If you delete CJUN from the cell line, then this behavior largely disappears. And entry into this CJUN program is also associated with mechanical differences in the Schwann cells. You can see in the bottom, these are AFM measurements that we did on live cells. And in the control, which is the CJUN reprogrammed cells, you can see that the stiffness is generally low, but in a CJUN knockout, the stiffness is much higher. And these stiffness differences are actually what explains the ability of the Schwann cells to adapt and push along these cancer cells. And so they came up with a model based off these microchannel data, based off Matrigel assays, based off a ton of IF experiments, done in conjunction with our core and with others. And they came up with this model where cancer cells, or PDAC cancer cells, run along these Schwann cell highways. So you have an encounter between cancer cells and Schwann cells, which are surrounding the nerves. The Schwann cells are in purple. This causes the Schwann cells to enter the CJUN transcriptional program, which is kind of analogous to what happens if there's a nervous injury. And then you get these tracks of Schwann cells building up, and you can see this in the kind of right two panels here. And there are actin differences which you can measure through microscopy and other methods. And these attract cancer cells, they bind cancer cells, and then as they move along, they bring cancer cells with them, and there's some active movement of the cancer cells also. And you can see this in the Matrigel models in the paper, which I encourage you to read, and also in live cell experiments in mouse also. They recapitulated all of this very nicely. And so you get this really, really nice interplay between the mechanical properties of normal cells and mechanical properties of cancer cells. You can measure those, you can visualize everything directly through light microscopy. It was a really fun project to do. It's continuing. We're doing larger scale AFM. We're doing a lot more confocal with them. Yeah, it's been a wonderful project. I don't know if anyone here has AFM in-house or is collaborating with other cores or other researchers for AFM. But if so, please reach out, and I'd love to talk shop and hear your experiences. But with that, I'd love to acknowledge the people that did the work in our facility, in the Wong Lab and others. Former colleagues Annalisa Callow and Lakshmi Gosain, who did a lot of work in sample prep and AFM. And thank you very much for listening. Thank you.
So Murray will be around for the whole meeting, so if you do have questions or you want to chat with him, please find him. So now we're going to switch to the activities of the LMRG. So for those of you who don't know me, I'm Rich Cole. I'm the chair of the LMRG for a little bit longer. Erica, I thought I saw you come in. Oh, there she is. So she has agreed to co-chair this wonderful endeavor going forward. So a little bit of background, because some of you aren't familiar with what project we're even talking about. So this was a worldwide image analysis challenge that we launched probably a couple years ago now. And it was both synthetic and real images. And we asked researchers, image processors, analytical people to basically analyze these images, provide data on how they did it, and then provide specific metrics. And the metrics differed from the different types of images and what we were looking for, whether they were C. elegans or nuclei. And it turned out the analysis to be a lot more complicated than I had hoped it would be. So the people that did the heavy lifting are like Thomas, Angel, and Chris, who's not here. Angel's not here either, is he? Okay, so I'm going to let Thomas explain some of the results that we got from this very complex image analysis challenge.
Jeez, sorry. Well, thanks, Rich, for the introduction. And yeah, I'd like to basically start with acknowledging that this is the LMRG group is quite a large group across the U.S. and a few people really helped in this. And Rich, thanks for trying to push us through and get the paper wrapped up. Michelle and Chris and others were helping right at the start with some of the questionnaires that you'll see. Angel and John helped with the image analysis, and David was basically right there at the start to do the synthetic image generation. So yeah, I'm just going to repeat some of what Rich already said. Apologies for that, but research group within the ABRF, roughly every year there's a new study. This is the 4th iteration. Now, number 4 study was to investigate results variability in image analysis. So the focus wasn't really to find what method delivers the best results, but it was rather, if you give the same problem to a number of researchers, how much is the variability that you get back in terms of the results that you get. And so we organized a global challenge. Sounds grand, but it means that it was open to anyone in the world, really, and generated artificially. We had artificially generated images because then we knew the ground truth, right? And we withheld the ground truth. We gave the artificially generated images, and we asked to perform simple analysis just to keep the audience as wide as possible. It was to identify nuclei in some HL60 simulated cells and do some spot counting in the C. elegans. Again, the goal was to investigate the variability of the results rather than ranking algorithms or software, which is usually the focus of a more traditional challenge. So we had 2 specimen types, HL60 and C. elegans. We were studying 2 components. One was the nucleus, and the other one was fluorescence in situ hybridization. We didn't specify, what protein, but essentially we had like spot detection into C. elegans. The images you see on the left are the C. elegans images, and the one on the right are the nuclei images. We had 2 signal to noise levels, and then 2 object density levels, meaning how close were the objects to each other. To generate the artificial images, we used Cytopack. It's a software written by the Center for Biomedical Image Analysis at Masaryk University in the Czech Republic. They had both a web interface and a command line. It's open source software. The web interface looks like this, and it's still up and running if you want to check it out. There's a bunch of different things you can change, and I've highlighted the nuclear images. The C. elegans was something that they developed specifically for this challenge. It was something that they didn't quite have something to detect those finer spots, and so we asked them to develop a module, and now it's integrated into the web interface. So if you go online, you can actually see it. You can also download the software. It's being containerized too, so if you have familiarity with any container running environment, you can just pull it from there. I'm happy to give more details to anyone who's interested. So this is how the specimens look like in 3D. This is the low clustering, high signal-to-noise ratio. On the left, the C. elegans with the spots, and on the right, you have the nuclei. And then this is the high clustering and low SNR. You can start seeing noise in the left image, but also the fact that the spots are also overlapping somewhat to each other. So a more challenging problem to solve. We also asked to analyze a calibration data set. This was mostly to deal with the fact that a lot of different software have different conventions. Not only even basic things like axis ordering. If you save the image, you might find the X and Y image axis flipped, for example. And so these were it was an which which had no background, 0 background, and a level, I think we used 255 just to make it more easier for software like ImageJ, which already have an idea of what the intensity for a mask should be. And so, and then it was 4 spheres. We had one at close to the origin, and then another sphere, then other 3 spheres, and each one was at a distance, was basically moved across along one of the axes at a particular distance, and which was different for each axis. So you could always, if you had those 4 numbers, you could always see, what was the order of the axis. And so we asked the participants to analyze this image too as part of their workflow. We're using the same software. It doesn't need to be the same parameters, but the same software. You would imagine that if you change the parameters of the software, the conventions don't change. It's more like the overall thing. Now I'm going to put this right up front. There is a known limitation to this study, and so when we generated the nuclei images, and when I say we, I say me, so blame me. Essentially, instead of the PSF that we had thought of, we kind of created for the project, it was a test PSF that made its way into the results, which means that the images were slightly better, higher resolution than the ones that for the nuclei, higher resolution than the one that you would expect. So on one side, easier to segment, but not corresponding to the metadata. We discovered the issue too late, so after receiving all of the participants' contributions, and however, I'd say that everyone was affected in the same way, and we don't believe this invalidates the study itself, but only the fact that the images might not be realistic in the sense that we would have liked to. And in particular, this comes up if you're trying to do deconvolution, right? So if you're doing deconvolution and trying to put in the parameters that you see, you will see your PSF being different from the pattern that you get. Okay, so in terms of the submission for the nuclei, we just asked for the position of those nuclei and the integrated intensity and the volume. For the C. elegans, in a similar way, we just asked for a table with the centroid locations, the X, Y, and Z. Columns should be named X, Y, and Z. Integrated intensity with a label intensity. Then, as part of the submission, we had a questionnaire. This was more to going into the details of how they got to that result. And so, for example, what software packages, any plugins, libraries that were used, what steps they took in the analysis at a high level, like names of any filters that they used, algorithms, and so on. Some justification of why they chose that particular strategy, how long it took to segment the image, and then, interestingly, how confident they were in the result, right? We also asked to self-evaluate their level of expertise of whoever was submitting the results, and also to upload some code or macro pipeline file, if applicable. Now, once we got the results back, essentially you get the list of a cloud of points, essentially, and you need to match this cloud of points with the ground truth. How to do that? Well, there are 2 family of ways to do this. One is using least sum assignment, least sum assignment, which is essentially if anyone is familiar with the Hungarian algorithm, it's essentially you take 2 point clouds and then assign to each point in one set 1 point in the other set. And then once you get this assignment, and it does this in a way to minimize if you take the sum of the distances of all of the assignments, it's done to minimize this sum. That's why it's called the least sum assignment. Once you have this assignment, then you can do statistics on those links, right? And you want to see how long they were, right? And the other thing you can do is also look at how many links you had and how many points were left out. And so you get essentially kind of a sensitivity and specificity of the algorithm, which was summed up in a Jacquard index, which kind of looks at the number of points in the intersection and divided by the number of points overall. The other family of doing this is without doing an assignment or not a specific assignment. You take each point and look at the closest neighbor and then do statistics on those links too. Okay, so now onto the results. First, in terms of demographics of participants, we had 29 participants from 6 countries. We had both participants from the public and private sector, both from academia and industry. By far the largest group were biologists, or self-identified as biologists, and most of them had a doctorate degree. Interesting enough, I'd say more than half of the people rated themselves in the top quartile in terms of expertise. And vast majority from core facility, which would make sense with the context of where this study was generated. Now, interestingly enough, a lot of the submission needed some help to kind of be analyzed, like things like conventions for the name of the columns or the file format for the things were not always respected, and so there was some massaging there. Well, not massaging, but just changing the name of the columns, order of the columns, and things like that. The other thing is that some of the scaling parameters were not applied correctly. So we asked the data in microns. And if you make a mistake in putting the scale inside, your results are completely off. And so it wouldn't make sense to analyze that because your result would be terrible. But again, it wasn't really interesting in itself. It's interesting in itself, but we were really trying to, okay, but that's not something that would, that's not what we're looking for. We're really looking at, once we imagine you got the scale right, what would be the variability in that, right? And so we really made an effort to go in and save the vast majority of data sets so that we could do some statistics on that. So what we did, we took the ground truth and we used a point cloud registration method, iterative closest point. And it's a standard for doing this. And then after registration, we did the linear sum assignment and the metrics calculation. Now, the anomaly as the axis ordering, you could detect in the calibration data, or you can also detect visually. You see if there's a mistake there because the image looks completely off. Also, errors in origin and scale were corrected using registration. Sometimes we saw software that had a 0 origin, a 1 origin, and also 0.5 origin too. So, of course, all of those have very good reasons. I was involved in a single molecule localization challenge a few years ago, and that's one place where you do see like the effect of like a half a pixel in terms of registration, because it's all a pixel is not really a square, but you can think of it as a square, right? And you can think of putting when you have index 0, 0, where is that? Is it like in the top left corner of the pixel, or is it in the middle, or not? MATLAB has one-based, indexing. Everyone else uses zero-based indexing. So things like that can appear. So here are the results, the effect of clustering and signal-to-noise ratio. You can see, expected trends that if you have higher clustering, the error is a bit higher. I should explain the axis. So the LSA is using the sum assignment. MSE is the mean squared error. So if you look at the mean squared error, when you have like low clustering, high clustering, you can see in the nuclei that you get, slightly different. Slightly worse. Not too much though, but slightly worse clustering. Same if the images are noisy, you get some worse localization. The effect is magnified in the fish signal, of course. Then, especially in the signal-to-noise ratio, you can see if there's noise, then it gets harder to detect those spots, and therefore you see a worse result. So let's see. So here we were looking at an interesting effect. So as I was saying before, we asked the participants to rate their what they would consider the level of expertise and how confident they were in the results, right? And then because we had the objective metric is to see, how well does that correlate. Interesting that there wasn't so much of a correlation between the two, which is interesting, and it kind of at some point makes sense. It's very difficult to self-evaluate, in a blind test like this. But kind of to make the point, maybe the take-home message here is that, try to be as objective as you can in terms of when you do your experiment. Try to put into place validations that can really make you more confident about your result in an objective way. There was a correlation between confidence and self-evaluated expertise. The more experts were more confident in their results. But again, given the fact that I just said that there was none of those who really correlated with the results, also means that if you've been doing this for a while, you're more likely to consider your results correct, even though they might not be actually. So that was an interesting result. Now this is a way we tried to visualize the fact that many people were using, well, everyone was using a different sequence of operations. So you see the actual operation that you're using in a different color. And then you had 4 images for the nuclei, and you see each one of those in a row. And then each participant is a different number there. So you can see, when you see this color here, that means that they used the same pipeline for all of the 4 images. So they didn't have to kind of copy-paste all of the steps in 4 times in the questionnaire. But you can see that most of the time people used the same approach to segment the 4 images. But sometimes like if you see like in this example here, one here, there was some adjustment done, according to the to how much clustering you got and how much noise you found in the image. Also to note that not all of the participants analyzed all of the images. That was also another thing that we noticed. And that was also, because we tried to reach as much people as possible, it was also said explicitly, if you don't need to analyze all of the images, because especially the questionnaire part, takes some time. So we were trying to make sure that we had a good pool of results, but if you had time for only one, that would still be useful. So these were for the fish images, similar pattern. Some of the people use the same pipeline for everything, but I would say more often than not, you would see small adjustments for depending on the condition, maybe adding an additional filtering step or something of that sort, like you can see in participants 21 and 37. Another way to visualize the sequence of steps is using these Sankey diagrams. So you can see essentially the flow from each step to the next is depicted here. So these are people who did like filtering and then thresholding and then maybe some outlier exclusion, and then the diagram ends here. So you can look at the different paths for all of this will be included in the papers, and these diagrams also take some time to kind of digest. This is the same for the fish images. At least there were fewer kind of overall number of pipelines, different pipelines, so you can see a bit more consistency here. Other findings, for example, we put in 2 buckets like simple thresholding and more sophisticated algorithm, and there was a lower error and the lower interanalyst variability when using just simple thresholding for the nuclei images, but the opposite was true for fish images. So maybe a take-home message is like simpler problems benefit simpler approaches. You can think of that if you start introducing methods that have more complex, they might be sensitive to more parameters, and therefore you might get a higher variability. Another thing that was interesting was that the variability between analysts was lower and had fewer outlier when using commercial software, which kind of the number A, the first one like A would make sense, right? Because typically in a commercial software you would be guided through the analysis, and therefore the workflows are kind of predefined, and therefore there's the least amount of variability in the actual result that you get at the end. And then the second one was that we saw fewer number of outliers in commercial software, which again might make sense because that outlier removal would be included into commercial software by default. So it's, they're interesting. Again, this was not a competition to find who's the best, but rather to see, what is the effect of these, all of these factors into when you're looking at different things. Okay, so concluding, we're still wrapping up the manuscript. There's plenty of things to see and further analyze. We could analyze the data for another year probably, but rather we're going to make everything available to anyone to dive into. There's might be some nuggets that we missed, but definitely want to make sure that everyone has a chance to look at the data. The data will be anonymized, so the name of participants and everything will be included, of course. And then the one conclusion is, of course, yes, you do actually get quite a bit of variability if you get the same problem to a number of participants. So anyway, thank you, and open to answering any questions.
We're going to ask you to use a microphone because there are a lot of people here, so I can either run around or people over there can go to that one. Okay.
Hi, this is, thank you so much. This is amazing work. So I know that the goal was not to make a challenge, but was there was there, how overall did it match the actual ground truth? I mean, for example, in the nuclei. I know that there was limited variability in the nuclear segmentation, but was it actually on the ground truth or somewhere else?
No, I'd say the vast majority. It was generally speaking like a simpler problem, like the nuclei identification. It was not hard in itself, and I'd say especially for the nuclei, the match with the ground truth was overall quite good, regardless of the actual pipeline. For the fish, the results were a bit more varied. You see more outliers. You start seeing some misdetections, and so clustering effects also. If the things are close together, you start to see subtle differences. And the other thing is that if you take even one software, especially in the open source world, they're designed to be as flexible as possible, meaning that you can use them in hundreds of different ways. And so even having like a ranking of, okay, well, this software, if you average, for example, all of the results from, say, Fiji, that includes a whole range of ways to analyze the data. And so that's, diving that. And also in a challenge, because the goal is different, you're kind of having a much tighter sets and much tighter control on the sample themselves and what you're looking for. So it's a different, it's a different thing. But I would say overall, everyone did a good job at doing that. If I might actually comment on that, one of the things that I think might be kind of a next step, if you like, is okay. So we got the error, but how good is that error? How does that relate to the actually answering the biological question? Because that's a hard one to frame in a challenge kind of way. But you can think of that if you had like a way of framing a challenge where you're saying, this is my biological questions. These are the images and the experiments that I took. Can I get to that answer? And then we would have kind of a step more after analyzing the data. Is okay. Well, what is that answer? And that would be interesting because then you could see how many of the participants actually got to the answer that we were looking for. But at this point, we're looking at absolute measures of errors.
Joe?
Rich? What were the main lessons you learned about designing this study? Because I've been on the LMRG tangentially for a number of years now, and I've seen the studies evolve. What are we going to take away from this to help design the next study better, right? So that we can execute it better, have higher participation, and things of that nature.
Yeah, I think I don't know. these are things that might, like, kind of my opinions, not the LMRGs, so take them with that. But things that might help is, like, for example, the questionnaire was quite extensive in the sense of it ended, because we were trying to capture a lot of metadata. That also meant that it took quite a bit of time to get to that point, and so participating was, investing quite a bit of time. So possibly having either breaking it up into kind of levels of participation or something that if you want to have like a quick thing that people can do and then submit the results, that would be one thing. Another thing is the standardization of the submissions. We got we had to really dive into the submitted material to kind of correct for things like that. I wonder if nowadays is something where you can paste the results in some kind of web form in a way that kind of standardizes the submissions might be interesting, or at least, for example, gives you immediate feedback. Oh, don't worry about this column, just give me those three. Something of those lines. It's a bit of a balance also. How much time do you want to spend like in the setup phase, to actually have like a sophisticated kind of web app to do all of that. Another way would be to, okay, well, I'm going to use one platform. Like for this study, for example, we could have said, well, let's use an online platform and just give a very finite set of different parameters that people can tune, and then have them work through the online app to get the result, and that would be also interesting, and that's much more controlled environment. But yeah, those are things that come to mind. I don't know. Yeah, it's hard.
So my boss asked me to tell him the results of this study in less than a minute, which is always challenging when you're speaking with admins. Hopefully there's no admins here. So basically, he said, tell me three things. So there were two things that did not surprise me, and there was one thing that did. The thing that surprised me was the lack of correlation between what you say your experience is, what you say, your confidence is, and what you actually get. Occam's razor applied. This, in simple cases, use simple solutions, right? I mean, that kind of makes sense. And the other thing, too, is that and I argue this when I try to get service contracts for my software, is that the software, commercial software, has been designed to help the user get the data and the results that they want, right? It does, I mean, we've all used like a Mars, and I have no commercial interest in a Mars or anything, but we've all used software like that, and it does kind of lead you down. And this showed that, yes, when you do, the average person, if they use commercial software, they're going to get a better result.
Yeah, I'd say that a lot of the effort in the open source community has been on designing, incorporating algorithms, there has been recently a push into increasing like usability. That's also, and I think maybe the next step would be to guide users through defined workflows. And Cell Profiler does some of that in the sense that provides kind of pipelines that are designed for particular problems, and then you can kind of navigate your way. Maybe one call to action is to think about ways we can bring that guided workflow into open source software. But again, as you were saying, a lot of the effort in these software vendors is really to make it accessible to users, and that's what you get essentially. Yeah.
So were there any? Oh, sorry.
Hi, Jenny Schaefer from Vanderbilt. Thank you. I was thinking about, it's interesting the variability of the data, but it seems to be almost 29 variations in how people did it as well. And so I wonder if there's a takeaway here, or if you can comment on, this makes me think, it gives me more confidence to tell my users there's no one right way. Mm-hmm. And especially with the data too, that people's confidence didn't correlate with the answer. So I guess if you could, was it 29 different, like none of them exactly matched the way that people did it?
Yeah.
And what that tells us about the number of ways to do it and that there isn't one right way.
I think what I like to take home is that it's not enough to think you've done it right, but you really need to make sure that what you get is what you're expecting to get. So that validation is really important. So kind of building in additional kind of controls on your experiment, or building in some kind of guardrails, so that if you get something that in the numbers that looks off, you can kind of detect it as much as possible. Yeah, definitely. There's no right way, in the sense that there's even if you take like nuclear, you can identify nuclears in many different ways, and so there's as long as you get to a good answer, that's and also, how do you define a good answer? Again, that goes back a bit onto the biological problem, right? If you're trying to solve, counting nuclei is a very different thing from, for example, measuring internuclei interactions. For example, if you think of proximity between cells, at that point you really need to be sure of the outlines of the cells. And actually, another level that we haven't even looked into was, we did ask for whoever could upload the masks from the segmentation, and so that's a whole new level of analyzing the data. And I think that would also give some interesting results, because you can think of cases in which you might get the center right, but the outline is completely off. And again, going back to the biological question, which one is the one that you want? Do you want the boundaries, or do you just want to get the center? So it's like with, again, single molecule localization microscopy. One thing is to identify the center of the spot. The other one is to, and you can do that even if the model that you're using for fitting is not your true PSF. You can get very close results even if that's not true. But if you're looking for more sophisticated detection, then at that point you need to have more sophisticated models.
So I have a question with regards to, so this was all basically a little bit of variability, but not like a different treatment where you would anticipate substantial differences in size between them.
Correct.
So would you anticipate, so the variability makes sense because they all process them differently, but would you anticipate if they had, say, another image that was a treatment, if the delta would be more reproducible?
You're exactly right, and that goes again to the point, the next level of this would be to look at an actual biological question. Are we actually able to detect the treatment differences? And then, because then that delta, becomes important, because for some problems, if the difference is huge, you're putting something that kills the cells, right? And then, your count goes down. You don't really need to have a very precise count to detect that. But if the effect is very small, then you need to invest more in your pipeline to be able to detect those differences a bit better. So there's not a, in a way, it's a difficult question to answer, because it depends on what treatment and what problem you were trying to solve.
So this also brings up a point which I try to drive home to my users, but especially to my students, is that you need to do controls. So we all think about controls when we're doing the imaging, right? Whether you're doing blocking or something like that, or a positive, negative control. But when you're developing a pipeline that you're going to use to analyze, thousands of images or even hundreds of images, you need to have a control. You need to put something in there that you know what the answer is going to be. So you're basically comparing it to the ground truth to be sure that what you're doing is actually doing what you think it's doing, and it's not just this black box.
Yep. Yeah, exactly.
Thank you. Kind of along the lines of the biological question and how much variability is relevant for the biological question, I think also thinking forward for adding the biological piece, it brings up the question of, if you have a larger number of images, so there's going to be more variability between the biological samples because of preparation or whatever, is it optimal to control your algorithm, so optimize your algorithm on your test set of images and apply it blanketly across the entire experimental set? Or one thing that we often run into, if there are differences within the experimental set from image to image that you can clearly see, are outliers from your test data, there are sort of different levels of comfort, at least among our users, in how much you would tweak your algorithm or your workflow to reflect what you can visually see as the variability in those different experimental images. So I think, you're concentrating on error within a fairly defined set of images, but obviously the more accurate you get on your test set, the less applicable your workflow, your algorithm might be, for a more variable experimental set.
Yep.
And I think we've run into issues where that becomes very obvious in the experimental set, and then you have to decide, would you rather have multiple algorithms and higher accuracy, which you almost have to do on an individual sample basis, or apply a more broad algorithm that is going to have higher error?
Yeah, I think, generally speaking, when I've worked with researchers, I try to have like one algorithm and apply it to all of the different sets because then you're kind of applying the same kind of analysis to all of these. But defining those parameters of the algorithm, that's where you can play with like things like your test set and validation set. So the test set is something that you're just checking your parameters against, and then validation set is something completely new, but like another experiment round. So the more you can invest into this step of the more you will be actually comfortable with whatever answer you get. But I'd say in generally speaking, keeping the same algorithm across the sets would be that would be the way I'd usually go.
Just do me a favor, hand it down.
Hi. One thing that I just wanted to, one comment I wanted to make about the usability thing, which is, this is really valuable to one can see it in different ways, saying, we should just go with commercial software. On the other hand, you were also mentioning that we don't really, often with commercial software, we don't know what actually was done, so how did they actually get the better results? But definitely the usability is a really important aspect. And, and there is a reason why in the open source field it tends to be a lot of just focusing on putting more and more algorithms versus actually the maintenance and the usability of each individual one. And so I think this one take-home message could be really use this data to point to the importance of the usability in the open source. Or at least the importance of transparency in the commercial. So we can use both fields, but one has to improve the usability, the other one maybe has to improve the transparency, and then we can find ourselves in a world in which both sides have advantages, but we minimize the disadvantages of both of them.
Yeah, that's much of what that was half of what I was just going to say. I think speaking on behalf of open source software, there's the statement was made earlier of like, well, clearly the proprietary stuff is better, but I think perhaps it performed better on this single task, but the black box issue in that like a user, in other tasks particularly where it's like automatically deconvolving things or reducing noise for you, like maybe that helps your segmentation, but if you're doing quantitative analysis, you need to know what's in that black box. So also, I'll just caveat, I have lots of strong feelings about lots of things, so I'll try not to monologue here, but another response that I also feel strongly about in terms of image analysis. I think it is really important to actually have a single workflow that handles your entire, all of your data. And one thing that a solution we've come to in our typical workflow is, you can only you can only manually test so many images, and we do that and get I think a pretty good basic pipeline, but then we'll actually run that on a subset across, so we're often in high content imaging, in our lab. And so we'll say, okay, show me a single image from every well across every plate. How did this perform in my segmentation? And then tile it all together. So we've implemented extra workflows to make it very easy to see, okay, yes, the thing that I tuned manually on a small subset is performing blanket quite well, or hey, actually it turns out I picked the very wrong subset, I did not tune it on a large enough subset, and when I see, at a sprinkling across the whole subset, or the whole data set, I clearly need to go back and retune. But actually building into our workflow the ability to quickly and easily just check performance of a single task has been very important for us in this important task of using a single workflow on all of your data. Again, I have a big soapbox about that, so please, happy to talk more, not right now.
Yeah, and I think I would like to really try to and step away from that dichotomy of open source versus commercial, because what we're trying to say, I think, is again, to Katarina's point, is there's a lot of investment done in usability and making it easier for users to go through the analysis, and that has a clear benefit. So that's one thing. And it doesn't matter if you're charging for that or not, but oftentimes it takes time and money to do that. So that's why that's an avenue that commercial software providers have. On the other side, there's open source software that is not reproducible. So it's really, it's not that, for example, a lot of papers are written with a link to their GitHub page, and that means it's open source. Well, yes, in a way, there's some code there. But then when you try and put it into practice, it doesn't work, impossible to use, or you apply it blindly because that's the parameters that have been baked in, and then you get some result, and you think you've done a good job, and then you get into errors. So I think there's a lot to learn from both sides of the aisle, and I think that's hopefully this might help to make that case.
So are there any other questions about this? We're all good? Okay, so we're going to switch topics here. All right. And we're going to go on to what the next RG project is. So which one do you plug in, Tom? All right. So, let's hope that this. Do you see my slides? I can't even see it from here. Okay, so we've had 4 or 5 very heavy lift projects in the LMRG. So it was basically, the idea was to let's do something a little bit differently this time. And so what, as a group, we picked was AI. And at first you may say, that's even worse than doing image analysis. So we have a different take on this, right? We're not going to put out, test sets and ask people to, develop neural networks and then basically, develop AIs for this. So different, very, very different take on this. So what we've already started is that we had a town hall. We started with a town hall on AI. It might have even been last year. And I expected maybe 20 to 50 people. Well, 500-700 people showed up for this. And we had 4 breakout rooms in this, which we thought we could, basically talk about stuff, everything from ChatGPT for helping training to, commercial software to, image analysis, to, basically image acquisition and AI. It was a complete disaster. Everyone was pissed. There wasn't enough time. People wanted to be in multiple sessions. But what it told us is that there was a general need for this and general knowledge. And one of the biggest takeaways that I got from this, from all of the emails that I got, some positive, a lot negative, was people did not know what AI was. They couldn't tell you AI from machine learning, from deep learning, and this is a problem, right? So AI is not going away. That's why I had Google, yesterday give you that very high 30,000-foot view. It's going to affect all of us in our professional life and in our personal life. It's here to stay. It's going to happen. So what we're doing is that we've had 2 of these. We're not calling them town halls anymore. We have to come up with a catchy name, which is not me. I need somebody who's into marketing. But we're going to have 6 more, right? And the first one is going to be on what is AI? What is machine learning? And with the idea behind these is that they're going to be able to give you take-home solutions, so that it's not going to just be somebody saying, well, this is it with bullet points. Is in a lot of them, we're going to provide, just like we did with the ChatGPT, data ahead of time, so you can look at the images. And then we're going to go through, the first one is like, all right, what are some examples? How do you actually implement this? What can you use it for? What can't you use it for? And it's going to progress, right? So that's the first one. We're going to have one, and this survey I'll talk about in a minute really pointed this out, on using AI for image acquisition, which to me is like one of the lower bars, but very, very few people do it. Then you're going to have the typical ones on, AI for image analysis, and we're going to split that down into, basically commercial software. So we're going to have a bunch of the companies that have commercial packages actually give their spiel. And then we're also going to have a session where it's like, write your own. And I realize that that's probably going to be the least popular. Most of us don't write our own, especially when you start talking about AI and you start talking about, developing these large models and stuff. When Google, and that's why I had him actually give some dollar values, which he had to get permission for, is it takes a lot of time to do it. And it takes a real lot of time to check it and to make it accurate. So one of the first things we did is we launched a survey. And when I say I launched a survey, put it out on the Confocal list server to basically get people's opinions, comfort levels, and where they were at with AI. And the idea is that, this was a very simple survey. I think it was 10 questions. Didn't ask a lot of information because we wanted people to fill it out, right? We know this from this project that the barrier, once you get over 3 or 4 minutes or 5 minutes, people stop doing it. This is known throughout the industry, especially if there's no economic incentive and we weren't giving out prizes or anything. So what I do want to do is kind of go through these questions, see if they shock you, because I know I don't have a whole lot of time. And then at the end, so probably by the end of the year, because we're going to do all these sessions, there'll be once a month. And the other thing which we found or thought of, and everyone has volunteered, even people who weren't there got volunteered, is after these sessions is we're going to have an office hour the following week, right? So what that does is that it gives you the maximum amount of time for the specific topic, and then the people that are really interested in it or really have questions or really want to dive in deeper can then join that basically that office hour the following week. And this gives an opportunity for, a deeper discussion. There's going to be way fewer people that do that, and that's okay, right? That was kind of the goal for it. So everyone can see that. So the very first question is what was your role? Your role in the core? And the vast majority is you're either the core director or core staff. So you see very few people in the AI are really kind of out of that realm. So how many instruments do you have in the core? As you can see, it varies tremendously, but there's a lot. And I should say that there was 115 people responded to this, and what also shocked me was 115 people completed it, that's also pretty unusual. So do you support image analysis in your core? Now this was to judge whether the core does image analysis. This was not about AI, right? And what you can see is that the majority either officially or unofficially support it. And anyone who wants these slides or wants this, just email me, and I'm happy to provide it. So that also is not very surprising. In this day and age, just taking a pretty picture doesn't cut it. You have to do analysis. But we wanted to set that as a baseline for this next question. Where is your comfort level with AI? And I don't think that this surprised me, but if you look at, I wouldn't consider myself an expert, it's the very largest one. So it's, on mine it looks, I'll call it yellow, right? It's the third one down. Even if you go up one, I have some understanding of this. And from the webinars, the people who say they have some understanding, the vast majority of those people really don't. I hate to say that, but even people who I know personally and some of the questions. So that's where we're at, right? This is the baseline. This is before the webinars. How often do you use AI in imaging? You can see where it falls, right? Not a lot of people are using it. This is the one that probably surprised me the most, is do you use AI in image acquisition? Over 70% said no. So I will tell you that I have no commercial interest in any software package. I will use names, but it doesn't mean that I'm recommending it, because I work for the Department of Health. I have to make that disclaimer because my boss could actually watch this. So a lot of software, and Nikon included, they have a little AI button at the top of their ribbon. And when I asked what this was such a large percentage, I knew some of the people that took this survey. I said, don't you ever use that? I said, have you ever clicked on that button? And the response was, no idea what it did, so we're never going to click on it. So that's why one of the sessions is going to be on AI for image acquisition, because clearly people don't understand it, because if you don't understand it, you're not going to use it, and you're not going to recommend it to users. I think that kind of makes sense. This is about using AI in analysis workflows. And although over 50% of the people said yes, when I went back and asked 6 of the people that said yes, they were using machine learning and they were not using AI. So again, this comes back to the point where this lack of understanding or distinction between AI and machine learning. And people will use those terms synonymous, including my boss. So do you use AI integration into commercial packages for your acquisition? Again, over 60% said no. And this is commercial packages, right? This isn't, you're not writing something. This is literally clicking a button in your software. And this was probably what surprised me the most. Do you use it for AI in your commercial package for analysis? So again, here, this is not too surprising. The fact that over 30% don't use it at all, and this is something that is basically going to become the norm, surprised me a little bit, but not so much. The outliers, so also you should look at, it looks green to me. I'm a bit colorblind, so I don't know, but the very top one, less than 8% just immediately said yes. Right, that's a really, really small percentage. And you can see it at the bottom where it's basically if you're using a non-commercial package. This also is not very surprising at all. So, do you use open source? And when we talk about open source here, and we've talked about open source for other image analysis products, this is, we considered this, and the description is any software that is not commercial, right? So this includes, all of those type of open software. And one of the things that I have experienced, and I will caution people, is because this is the wild, wild west, and this is a new frontier, is if you're getting stuff off of GitHub, you better have some controls that you're putting through that to make sure that what you're getting out is real. Some of the models that these are built on are, I hate to use the word flawed, but are maybe not as good as they should be, and some of the embedded parameters in there that which you do not have access to also may be incorrect. So this is equivalent to when ImageJ, you guys are all probably too young, but when ImageJ first came out and everyone was writing for it, and we found out a lot of the stuff was not accurate. So that's just a word of caution for that. And this didn't surprise anyone. You generate your own deep learning, machine learning pipelines. So this is basically where the bar is kind of the highest, where you're actually developing these things. And we saw yesterday that it takes a lot of resources to do this. It takes really a dedicated person. And the other thing too that I'll say, and it came from this image analysis challenge, is that it may not always be necessary, right? Occam's razor applies. We saw it in this. It applies in AI also. Having the most complex models, having the most complex pipeline doesn't necessarily guarantee you better results. Sometimes it will actually give you worse results. So that's not surprising. And it's also not surprising that almost 80% of the people said that they don't do this. So if you do not work with AI, which was the large percentage, do you expect to work with it in the future? And this is what kind of drove us to do these series, is because we broke it down to 6 months and a year, and you see the vast majority of people, the vast majority, intend on working with AI in the next year. So that's the top two blocks. So that says that basically people are going to do this whether they kind of know it or not. And I think because it's expected. And I know from talking to other core people and stuff is that the users, our researchers, they go to conferences, they read journal articles, and it's just like when, the latest, greatest microscope came out, like when super resolution came out, they all want to do it. No idea what it is. No idea if it's suitable to their research or their project, but they saw it in a paper, their colleague did it, they want to do it. So there is that pressure to, and at least you want to have the understanding to say, no, you really don't need to use a STED microscope when you're looking and counting nuclei. And that actually, I've had to say that to a researcher. You should be on a wide field microscope. You do not need to use a STED because you're counting nuclei. And then we had some demographic information. So I know I'm just about out of time here. Are there any questions, comments, thoughts?
Hey, Rich.
Yes.
Very nice. I just want to give a little bit of a quibble and then a little bit of a advertisement. So the quibble is technically ML, depending on how you define, like if computer science-wise, AI contains ML, contains deep learning. So in that sense, people doing machine learning, that is people doing AI, depending on how you define it. That's typically the computer science definition. But the other sort of the advertisement would be Thomas and Kevin Elisari and I are going to be doing a thing about incorporating AI into machine learning core or into cores on Wednesday afternoon. So definitely recommend folks come to that session. And we are going to talk about ways that, or I'm going to at least highlight one of our tools for training your own machine learning models with no code. So there are starting to become tools for this, but yeah, it's definitely a thing we all need to talk about more, we all need to think about more. So thanks for presenting this awesome data.
So Beth, what time and what room?
It's Wednesday, 3:30. That is a good question. What room? Yeah.
Just so that people.
Yeah. I should have looked it up before I came up here, but yes. Greenway. Oh, 2 o'clock, sorry. Yeah. Yeah, so clearly I know lots, but I do know more about machine learning than I do about actually keeping my schedule together. But yeah, Thomas and Kevin and I will be talking all about this, so.
Okay, so it's 2 o'clock.
2 o'clock on Wednesday in Greenway.
Okay, any other questions, comments? Because I know I'm keeping you guys from lunch. Okay. Well, thank you all for attending. Thank you guys if you were the ones who participated in the survey, and participate again.
Looking for someone, that perfect girl, who can take our hand and help us see the world. But I'm a little unusual, because you see,